Hey there, welcome to Chapter 30, An Introduction to Animals. So by the end of this chapter, you should be able to tell me why an animal is an animal, to be able to describe the key innovations to body plan that occurred during the early radiation of animals in evolutionary history, place the major lineages of animals correctly on a phylogenetic tree, describe how animals within lineages have diversified in things like how they sense, feed, move, and reproduce. All right, so where do we leave off? We talked about the fact that the ancestors to animals were single-celled protists. Okay, so this tree we already looked at before, I think. So this is the clade that is eukaryotes, and within the eukaryotes are several lineages of protists, right? And then that one clade, the Opisthokonta, gave rise to the lineage of animals, okay? So protists are a paraphyletic group. Animals, though, are just this one branch. They're a monophyletic clade. Animals are eukaryotes that share three key traits with one another. They are all multicellular, so there's no unicellular animals. They all have cells that do not have cell walls, okay? So they have cell membranes, right? But they don't have hard cell walls on the outside. Instead, their cells have a membrane that's surrounded by an extensive extracellular matrix. So if you can kind of picture back from 1113, it's like that kind of big old garbage dump of different kinds of molecules that surrounds the outside of an animal cell. All right, the second thing that animals have is they're all heterotrophs. So they all have to get the necessary carbon compounds that they need from other organisms. And so none of them are able to make their own food. Most animals ingest their food rather than absorbing it. So they take food into their body. There are a couple that just kind of absorb stuff through their body wall, so we'll get to them. The third characteristic is that they're all motile, meaning at least at some point in their life cycle, they have the power to move on their own. Okay, so there's a lot of animal phyla. We're not going to cover all of them. So there's currently somewhere between 30 or 35 animal phyla, depending on, how you lump them or group them. But people are constantly debating which are which. So we're going to cover a lot of these, but, for example, we're not going to be looking at the pharonids, the horseshoe worms, or some of these other weirdos here. The gastrotrix, they're super cute. There's a lot of them, but we're probably not going to talk about them. But we'll cover a lot of the other ones. Okay, so we have different kinds of data that we use to figure out what key innovations there were in each of the animal phyla. So the first, we have fossil evidence. So by looking at the fossil record, we can figure out what ancient animals looked like, when they lived, where they lived, and we know that not all animals are equally represented in the fossil records. We already talked about this. Does anyone remember which kinds of animals are most likely to get preserved in the fossil record? Anyone? Dog, do you remember?
Yes, things with hard bones.
Well, I know you like bones, but it doesn't have to be bones, but it does have to be hard, right? It could be a hard shell.
Yeah, like it has to be a hard shell or a bone or something.
Yeah, or like pollen with a hard crust, right? So something hard. It has to have been an animal that lived for a long time and didn't just, here and gone. Something that was found throughout the world, something that was found in large numbers, something that lived in the sediment would be more likely to be preserved, and something that lived in a place where after it died, it would get covered up real fast. All right, the second kind of evidence we use, it comes from comparative morphology. And so morphology just refers to, kind of anatomy. So this is comparing shared characteristics and synapomorphies in anatomy, and we can use this to define the body plan, meaning, I don't know, stuff like how many legs you got, you know what I mean? How many antennae do you have? Just kind of structural aspects of how animals are organized. And then we can infer which characteristics arose first and which animal groups are more closely related to each other than which ones. The third thing we can use is comparative development. So we can see from the time that you have a fertilized egg how the different blocks of cells grow into, kind of different structures. So we can look at how, for example, genes are expressed and how that affects the developmental pattern of the cells. So we know that sometimes really diverse structures on animals can actually be determined by similar genes and developmental processes. So that's one thing we found out. Conversely, sometimes you have structures that are very similar and do something very similar in different organisms, but they're actually determined by different genes and completely different developmental processes. So we need to have some information about comparative development to determine relatedness among groups. The fourth thing is comparative genomics. And so, do you remember what the genome is?
Yeah, that's just like all of the DNA you got in your entire body, like coding, non-coding, like all of it.
Yeah, so that's genomics. So we can use all of that information about genes and genomes of diverse organisms and see how they're similar or different. And so lately, now that we can sequence whole genomes of many, many organisms very quickly, we have learned a lot about phylogenetic relationships and evolutionary history by doing this kind of comparative genomics. So a lot of the trees that I'm showing you in this chapter and in the next chapter are things that we have just figured out within the last 5 years. Okay, as I said before, animals are monophyletic. So, all right. So this clade here from here on out are the animals. Do you remember what choanoflagellates were? We talked about them a little bit before.
Yeah, I remember those guys. Those were like protists that were like had like a little collar and flagella on them that looked like sponge cells.
Yeah, so that's what a choanoflagellate is. Good job. All right, so all animals have a single common ancestor, right? So the first group we're going to talk about are porifera, sponges, right? Like they have pores, right? Porifera. And so porifera include two lineages that are basal. And so we think that multicellularity originated in some kind of a sponge-like animal. Dun dun dun, or did it? We'll come back to that later. But one thing that we've thought for a long time is that sponges are the basal animal in the clade of animals, and that the ancestor of all the other animals was a sponge-like animal. All right. So look at this tree here. Are sponges a monophyletic group?
Oh, no, they ain't a monophyletic group. Cause like there's two different branches and the common ancestor of both of the branches includes the whole rest of the tree.
Yeah, see? The common ancestor of this sponge and this sponge would be here, right? So a monophyletic group, what's the definition of a monophyletic group?
An ancestor and all of its descendants.
Yeah, so if this is the ancestor, all of its descendants would be the whole tree, right? So what do you call a group that has some of the members and then kicks out some of the other members? Remember what that one is?
That's a polyphyletic group.
Yeah. Okay, so we have some fossil evidence that supports the idea that the first animals were sponges. So the earliest fossils that we have found are sponges. So those date to over 700 million years ago. Okay. Sponges share characteristics with a kind of a protist called a choanoflagellate. Okay, that's what the choanoflagellate looks like. It's got a little kind of collar and then flagella that sticks out. This is what a cell that is part of a sponge looks like. We call it a choanocyte. Choano just means collar, so it's got like a collar. They both got a collar. Both sponges and choanoflagellates are benthic. So when you hear the word benthic, it just means that they live on the bottom of an aquatic environment, like they live on a lake bottom or an ocean floor. Both sponges and choanoflagellates are sessile. So sessile means that they're permanently attached to a substrate and they don't move. Okay. Both feed using very morphologically identical cells, this one and this one. And the way that they work is they beat the flagella, and there's a lot of them together in a colony, either in a choanoflagellate or in a sponge. And so they beat all their flagella together, and it creates a current. In this case of the sponge, it pulls water in through the little holes on the side of the sponge and out the big hole in the top. In terms of a choanoflagellate, it just draws the water up from below, and then the food particles go past other cells that are able to trap the debris and eat it. And so the feeding will occur at the cellular level. A cell is going to engulf that food. So there's no tissues, right? All the cells are separate. Okay, so there's also morphological evidence for the sponge's first hypothesis. So the choanoflagellates will form colonies, groups of attached individuals. One thing that sponges can do that no other animals can do is they can reaggregate. So if you separate the cells from each other, they can come back together again and form a living thing, right? So if you took any other kind of an animal and pushed it through like a colander or something or a mesh, that animal would just be smushed and would be dead. But if you do it to a sponge, all those cells can come back together again and reform because a sponge is organized at the level of the cell, cells working together, but not at the level of a tissue. Sponges, unlike choanoflagellates, have different cell types, and the cells are dependent on each other, so each of those cell types can't live on its own. And so the cells live in organized layers inside of an extracellular matrix, which is common to all animal cells. Some sponges even have something that is like a true epithelium, so they kind of have like a skin tissue. And so this is when the cells come very close together and form a tight layer that covers the interior and the exterior surface. Sponges are very diverse. Some are very large, some are tiny, they're made out of different things. So some of them have a skeleton made of spicules that are made of silica or glass, so the skeleton is glass-like. Some of them have a skeleton of spicules that are made of calcium carbonate, so kind of chalky. And so we can actually go through and look at the type of spicules to figure out the different lineages of sponges. All right, check this out. So we've taken cells from a sponge and separated them, and then if you leave them over long periods of time, they all come back together again to form a new sponge. So there we go, separated, time lapse several days. You can see all of those cells finding each other and starting to form the sponge again. Okay? If we look at the comparative genomic studies that have sequenced sponges and other organisms, we know that sponges are paraphyletic. So remember, there were multiple lineages of sponges. Those sponges share developmental toolkit genes that you need to do all of the basic molecular processes, even though some of those sponges aren't actually using genes to do these things. They could do them if those genes were tweaked a little bit. So they have genes that would allow them to specialize in cell types, which sponges do a little bit, to regulate cell cycling and growth, so to figure out when cells should reproduce or not, adhesion, cells sticking to each other, recognition of self and non-self, so the sponge can tell which cells belong to the sponge and which cells don't, so that's the basis of innate immunity. Developmental signaling and gene regulation. So this is what multicellular organisms use to be able to figure out, basically as they're developing, what part of the body they are, and so what fate that group of cells should have. And also program cell death. So when cells should die, either to produce structures or to avoid accumulating mutations. An alternative view to the sponge's first view is that ctenophores are actually the oldest animals. And there is new genomic data that has produced this alternative hypothesis. And so people have found that ctenophores are a sister group to all the other animals. And one reason that they think this is that ctenophores are the only animal that lacks microRNAs. So, do you remember what microRNAs are?
Well, they're made of RNA, and they're probably small.
Yeah, they're small. They're made of RNA. Do you remember what they can do?
Yeah, like they're like little scraps of RNA that can kind of get in there and like prevent translation sometimes.
Yeah, so sometimes microRNAs can get in there and modify gene expression. And it's something that's been found in all other animals, but not the ctenophores. And so it's kind of possible, it's possible that ctenophores had an animal ancestor that had microRNAs and that trait was lost, but that's kind of hard to picture. So we don't yet have sufficient evidence to reject the sponge's first hypothesis, but I think that there's very strong evidence that ctenophora may actually be the oldest animals. And that would suggest that the sponges lost their tissue level of organization later, rather than starting out never having a tissue level of organization. Okay, so biologists once considered sponges to be colonies of single-celled protists because sponge cells are able to reaggregate after being dissociated. What evidence may have helped them to realize that sponges are actually animals and not colonial protists? All right, should I read out answers?
Yes, please.
All right. Their feeding cells are flagellated.
Nah, that can't be it, because, like, you got protists with the feeding cells that are flagellated.
Yeah. They are suspension feeders.
No, you got lots of protists that are suspension feeders.
They exhibit radial symmetry.
But do they?
Yeah, I don't know that sponges actually exhibit radial symmetry. What else? They produce spicules made of silica or calcium carbonate.
I mean, they do that, but how would that tell you whether or not they're protists?
That's true. They have an extracellular matrix surrounding their cells.
Oh, that's the right answer.
How do you know that?
Because that's a property that all animals share.
Yep, you're right. The extracellular matrix surrounding the cells is a property that all animals share. So it's a synapomorphy of animals. Okay, so other than sponges, animals are divided into two groups based on the number of embryonic germ layers that they have. So we call them germ layers when they're first kind of developing in the embryo, and then later we call those layers of tissue when they're mature. So they could be diploblasts. So what's di mean?
It means two.
Yeah, it means two. So diploblasts have two germ layers. The ectoderm just means outside skin, and the endoderm, inside skin. Triploblasts have three germ layers. So the ectoderm is on the outside, the endoderm is on the inside, and the mesoderm gives rise to the circulatory system, so it's in between the ecto and the endoderm. So the ectoderm gives rise to the skin and the nervous system, the endoderm gives rise to the digestive tract lining, and the mesoderm gives rise to the circulatory system, the muscle, and internal structures like bones and organs. Alright, there are two groups of diploblasts, the ctenophora, or the comb jellies, which we've just seen, and the Cnidaria. So the Cnidaria include jellyfish, corals, sea pens, hydra, and anemones. But people have been kind of looking carefully at both of these groups, and they have found that some of the ctenophores actually have mesodermal cells in their mesoglia. So mesoglia is like kind of a jelly-like substance between the two, the ectoderm and the endoderm that you find in diploblasts. So like they have two layers of tissue and then goo in between, right? But some scientists have found that there's mesodermal cells kind of floating in the goo. Also, some ctenophores and cnidarians contain genes coding for structural components of mesodermal cells, but not the mesodermal specification genes. So it's like they have part of the genes that they need in order to make mesoderm, but they are missing some of the cells that they would need in order to enact that. So they don't quite have the ability to make mesoderm, but they have some evidence of genes that could be used to make mesoderm. And so ctenophores and cnidarians, we'd say, share some of the genetic tools for triploblasty, but not others. And then there's some similarities among the mesodermal cells in diploblasts and the mesoderm of triploblasts that may be due to convergent evolution. So some of the genetic material may be homologous, but maybe some of the cellular similarities may be analogous. So it's kind of a mix. All right, so what about muscle, right? We know that ctenophores and cnidarians, like they're moving, right? You can see them swimming. So how can they possibly change shape without that kind of contractile tissue or true mesodermal muscle, right? They don't have mesoderm. How do they have muscle? And so we know that all animals share these homologous genes for the contractile proteins actin and myosin. Do you remember those from BIOL 11-13?
Oh, do I ever? Like, there's the one that's like the bead, and the other one looks like golf clubs, and they like scrunch around next to each other, and then they make muscles move with the thin lines and the thick lines.
Oh yeah, you remember quite a bit about it. Okay, so that's actin and myosin. So all animals share homologous genes for those proteins that make up muscles. So ctenophore and cnidarian contractile cell proteins, they function like true mesodermal muscle cells, but they evolved independently. So they have the proteins, but they inherited the genes for the proteins from a common ancestor, but not like the full like muscles themselves, right? So they independently evolved a different kind of muscle cells using the same proteins. Okay, so some of the protein components of the muscle contractile apparatus evolved before you had 3 layers of tissue, before triploblasty. And so we think that the proteins that were present in ctenophores and cnidarians, and then also in triploblasts, were co-opted and elaborated upon differently in the different lineages, right? So the functional similarity and how muscles seem to work in the different groups probably happened by convergent evolution, okay? So the genes for actin and myosin are homologous, but the muscle cells themselves, the structure of them, evolved independently in the different lineages, so they are analogous. Okay, so before animals were bilaterally symmetrical, meaning the same on left and right side, they were radially symmetrical. So cnidarians, ctenophores, and some sponges have radial symmetry. Okay. Somebody want to describe radial symmetry?
Yeah, it's like if like a cake and you cut it and everyone gets a slice that's the same shape.
Yep, it's like a cake. If you cut it the right way, yep, everyone gets the same kind of slice. Okay. So animals with bilateral symmetry are sometimes called bilaterians. Okay. And all of the bilaterians are triploblastic, and yeah, so bilaterians are the clade of animals that are triploblastic, bilaterally symmetrical animals. There are some animals that are within this clade that have radial symmetry, and that would be the echinoderms, like the sea stars and the sea urchins and stuff. But we think that the radial symmetry in that lineage evolved independently later. It is interesting, though, that we tend to think of cnidarians as having radial symmetry. However, they have some components of bilateral symmetry if you look at their internal organs. The symmetry in bilaterians is due to particular genes. Do you remember Hox genes? You know what those are?
Those are those master controller genes.
Yeah, what kind of stuff do Hox genes control?
Like the timing of development.
Yeah. So Hox genes are responsible for regulating development of the anterior-posterior axis, meaning what part is going to develop into the head and what part is going to develop into the butt. Okay? There's a second kind of gene, I can never pronounce this correctly, the decapentaplegic genes, DPP genes, that regulate the dorsal ventral axis. That means those genes regulate which part is going to become the back and which part is going to become the belly. Okay? So the symmetry in bilaterians is due to the action of the Hox genes giving them a head end and a butt end, and the DPP genes giving them a back side and a front side. So some parts of the genetic toolkit for bilateral symmetry was clearly there even before the cnidarians and the bilaterian lineages split off from the other lineages. And so we think that's why you do see some bilateral symmetry in internal organs in cnidarians. And so we would say that that symmetry is homologous because it's due to the same genes in bilaterians as it is in cnidarians. But there's other parts of the body that evolved later after the lineages split, and in that case we'd say that there's convergent evolution for bilateral symmetry. So again, it's complicated and it depends on the specific structure. Alright, so here is a classic experiment that somebody did, trying to address the question, is bilateral symmetry in sea anemones homologous or analogous to bilateral symmetry in bilaterians? And so what they're looking at is, they're looking at the embryos of sea anemones. So when they're embryos, they look like they have bilateral symmetry, and they wanted to know whether that symmetry is homologous in sea anemones, and bilaterians. The null hypothesis is that bilateral symmetry arose independently in sea anemones and bilaterians, meaning the symmetry is analogous. So what they did is they stained the proteins that are produced by the Hox gene in developing nematostella, which is a sea anemone, embryos, and larvae to figure out where they get expressed, okay? So this is an embryo, and if you look at the embryo, the star is where the mouth is going to be, okay? And so this is the larvae, and then they treat it with this Hox stain, And then they also treat it with a DPP gene product. So do you remember what the Hox tells you? Or what the Hox gene controls?
Yeah, where the head is and where the butt is.
Yeah, and what's DPP control?
Where is the back and where is the belly?
Okay. So, the first question is, what predictions would you make if the main hypothesis is supported? And what would you predict if the null hypothesis is supported? And to help kind of guide you here, let's just say that the Hox genes we'll just do it with colors, where they express kind of a blue at the head end and a purple, right, at the foot end, okay? So where's the blue expressed and where's the purple expressed? And then for the DPP genes, let's just say that they give you, they express kind of a red on the dorsal side and a yellow on the ventral side, right? Okay. So, if bilateral symmetry is homologous in sea anemones and people, what would you expect to see if you stain that larvae with Hox stain?
Wait, what?
Well, you know where the mouth is, right?
Yeah, I see that mouth.
Okay. So, if the mouth is being produced by the same mechanism that our mouth is being produced, what color should the genes be expressed near the mouth?
Oh, like that, like blue-green.
Yeah. And what color should the genes be being expressed at the butt end?
Purple.
Okay. That's pretty good, given you can't see color.
I just remembered, colors don't mean anything to me, but, I humor you.
Okay. Alright. Anyone see color?
Yeah, I can see color.
Okay. So what about the DPP genes? Which would be the dorsal and which would be the ventral side?
Oh, I can't tell that part. I can tell what the mouth is.
Well, let's pick one side. Let's say the top is the dorsal side and the bottom is the ventral side.
Oh, then the top is going to be red and the bottom's going to be yellow.
Okay. Alright, so that's what we would expect to see if the bilateral symmetry is homologous in sea anemones and bilaterians like us. Alright, and what do we find here? Alright, so this is the Hox genes, right? So did we see that color, that blue color like we see in a human head near the mouth?
Yeah.
And then the purple color near the butt?
Yeah.
Okay, so does that look like it's being expressed homologously?
Yeah, it looks the same as with a person picture.
Yeah. What about the DPP genes? Do you see the one side having like the red dorsal stuff and the other side having the yellow ventral stuff?
Yeah, it looks the same. Hey, look, it's being expressed, homologously.
Yep. So this was a kind of experiment that led people to believe that bilateral symmetry is homologous in Nematostella and bilaterians because it's being controlled by the same genes operating in the same way in the sea anemone as it is in, bilaterians like humans. All right, let's talk about the nervous system. So sponges don't have nerve cells, right? So that's a 0. Cnidarians and ctenophores have nerve nets, so they look like this. It's a diffuse net of neurons. Ctenophores have a ganglia, so one ganglion, plural ganglia, and that's a cluster of nerves, kind of at a particular spot. So these are different kind of ganglia, right? Bilaterians have different things going on. Some of them have a nerve net, and some of them have an actual central nervous system, where they have a cerebral ganglion right here, and then they have clustered neurons that go down the body. So worms also kind of have a head, right? Evolving a head is what's known as cephalization, right? It means getting a head. And so people have noticed that animals that have a head also tend to have a central nervous system. Animals with a central nervous system also seem to have a head. So those two traits seem to have evolved together. And so one thought is that if you have a head, it's good to have a central kind of cerebral ganglion or a brain to be able to process information. So stuff is coming in. If you have a head, you probably have sensory organs on your head, right? And you're taking in information, and that needs to be processed, and that information needs to be sent to the right part of the body, right? Like, see predator, run away, or see food, eat food. Okay? So the head serves as a structure that concentrates all of the things that you need in one place for feeding, sensing, and processing information. Okay, so the basic bilaterian body plan is a tube within a tube, okay? So remember we talked about how there's an ectoderm and an endoderm, right? So the endoderm is this kind of a tube on the inside, and that tube forms the walls of the gut, okay? So that's the endoderm. The ectoderm forms the skin around the outside, right? You probably have mesoderm between those two things, but we'll not worry about the mesoderm for now. So just think, we're basically a tube within a tube, right? One end of the ectoderm has a mouth on it, the other end has an anus, right? All right, so the inner tube is the gut, with the mouth and the anus on opposite ends. The outer tube will form the nervous system and the skin, or on the outside, the blue part, and then the mesoderm in between will form the muscles and the organs. So a lot of animals have a coelom, which is an enclosed fluid-filled body cavity that's between the inner and outer tube. So imagine there's space between the inner and outer tube. And this can be really useful. You can use it to have some oxygen and nutrients that circulate throughout the body. Maybe just a little extra space so your independent organs can move independently of each other, right? So if you kind of bend over, you don't want all of your organs like contained exactly in their exact spot. You want them to be able to shift so that you can move your body. So we can imagine here's the blue nervous system around the outside, and then on the inside between the two layers you have your muscles and organs made out of the mesoderm. Okay, so we break up, we can kind of divide up animals into three groups based on what kind of a coelom they have. So a true coelomate is an animal, so by the way, I should say, you have to have 3 layers to even start talking about a coelom, right? So when we're talking about whether something has a coelom or not, it has to be a triploblast in order to even consider this possibility, okay? Anyway, so a true coelomate is a bilaterian, meaning a bilaterally symmetrical organism, whose coelom is completely lined with mesoderm. So the opening in the body cavity, this is a cross-section, has opened up within the red part, okay? Okay. So it's not in the very center of the body. This hole over here is the inside of your gut, right? So that's filled with food, or poop, I guess, depending on how high up or down you are in the gut, okay? We're talking about a different opening that is within the mesoderm. Like in humans, for example, our body cavity, like if you do surgery on someone and open them up and their organs are just kind of in there, right? Like their organs aren't, I don't know how to put it, their organs are loose, right, in the body. They're attached to things, but they're not like humans aren't like a solid packed, case with each organ in its compartment, right? And so like our body cavity is produced in a way that is surrounded by mesoderm and all of our organs are in that, right? All right, that's a true coelom. An acoelomate has no enclosed body cavity, so you can see there's their gut, right? But we're not talking about that hole. We're looking over here. So they're just like one solid chunk, right? There's no holes in their mesoderm or anywhere else. So the flatworms would be an example of an acoelomate. Pseudocoelomates, and again, we always give names like that we're like the real thing, and then this other group's like the bad thing, based on how similar or different they are from like humans or whatever. But anyway, so pseudocoelomates have a coelom, but that coelom is only partially lined with mesoderm. So you can see here, this gap here, on the one side it touches this mesoderm, and on the other side it touches the gut, okay? So the mesoderm doesn't go all the way around the coelom. The coelom is like, gut on one side and mesoderm on the other. So roundworms like nematodes and rotifers are both pseudocoelomates. All right, so if you look at the morphological data, it suggests some gradual evolution from simple acoelomates to pseudocoelomates to coelomates. However, if you look at the molecular data, so like when I was in school, we learned that whether something had a coelom or not was really important, right, for deciding where it was evolutionarily. We know now, though, that the coelom arose in some ancestral bilaterian, but then in each different lineage it could be modified, reduced, or even lost, okay? So if you figure out where the coelom is found here, so for example, we think maybe the coelom was first acquired here, and then it's lost over here. I don't think I have all the things written on here, but so these are pseudocoelomates, these are coelomates over here, coelomate, coelomate, pseudocoelomate, no coelom, right, coelomate, coelomate. So it's not a very useful synapomorphy for figuring out the relationship between bilaterian animals because it's very evolutionarily flexible, okay? It seems like it has been quite easy for different lineages to evolve different kinds of coeloms. Okay, so let's just say I gave you a worm-like animal to dissect during biology class.
Can I eat it?
Well, bird, you can eat it if it's not preserved in anything.
Okay, I'll wait till after class.
But you are not told what kind of animal it is. So you note that it has a fluid-filled body cavity that is partially, but not entirely surrounded by muscle. You infer that you must be looking at a member of...
Wait, what?
Well, I'll point you out to the part that you say, the thing that you note. So you note that it has a fluid-filled body cavity that is partially, but not entirely surrounded by muscle.
That means it's a pseudocoelomate.
Oh yeah, the pseudocoelomate.
Okay, so do you remember which of these is a pseudocoelomate?
Yeah, phylum Nematoda.
Yes, phylum Nematoda. Yep. Cool. Extra bonus. I mentioned another phylum that's a pseudocoelomate. Do you remember what it was?
Yeah, it was the rotifers.
Yeah, rotifers. There's actually a lot of different phyla that are pseudocoelomates, but because we're not covering all of the phyla that exist, we're only going to be talking about, I think, nematodes mostly. Okay. So, we have another way of distinguishing between lineages in terms of grouping them as protostomes and deuterostomes, so we need to talk about that grouping. So let's start at the beginning. So first you were an egg, right? And then it got fertilized, and then it was one cell, and then it divided and became 2 cells, and then it divided and became 4 cells, and then 8 cells, then 16 cells, then 32 cells, and then a lot more cells. And then the cells started to migrate from the center to the outside, and it became a hollow ball of cells, okay? And then at some point, that hollow ball of cells gets a little pushed in on one side, okay? So it gets a little indentation right here. And so that indentation is going to become part of the gut, okay? So that indentation will get pushed in and pushed in and pushed in until it goes all the way through and comes out the other side. So that first little hole could either become the mouth or the anus, right? So for deuterostomes, that first pore becomes the anus and the mouth forms later. And this name, instead of it being called like proto-anus, which would seem like the first hole becomes the anus, deuterostome just means the second hole becomes the mouth because they're too polite to call it proto-anus or whatever. So the protostomes, like traditionally when I was in school, we learned that first indentation becomes the mouth and then the anus forms on the other side. But we now know that development is actually really variable in protostomes. So in protostomes, this blastopore might become the mouth, the anus, both, neither, okay? So we no longer consider that this grouping protostome is super useful. For deuterostomes, this bit is true though. The first pore will become the anus and the mouth will form later. There's a second kind of a thing that I had to learn in school, and I'm not going to make you learn because we don't believe it anymore. So traditionally, we were all taught that the mesoderm was formed by different developmental pathways in protostomes and deuterostomes, like whether it was formed in blocks here and blocks there. So I had to memorize that when I was in school, but you don't have to memorize that because we know that this is no longer a good way to be able to categorize protostomes and deuterostomes because mesoderm forms in different ways in both groups. So if you're interested in development, feel free to learn about it, but it won't help us figure out how animals are related to each other. Okay, so phylogenetic analyses consistently support 2 major groups within the protostomes, okay? One grouping is called Lophotrochozoa, so that includes like the mollusks, the annelids, the flatworms, and the rotifers. And so this is a group in which all of the the different species grow continuously, so long as they get enough to eat and whatever, okay? The ecdysozoans include the arthropods and nematodes and a bunch of other groups, and they tend to grow by shedding their external skeletons or outer coverings and expanding their bodies. So they don't grow continuously, they kind of grow in jumps, right? So they'll shed their skin and grow, and then stay the same size, and then shed their skin and be a little bit bigger, etc. Okay, we also need to talk about segmentation. So when I talk about segmentation, I mean dividing a body or a part of the body into a series of similar structures. And so we know that the toolkit genes to allow for segmentation arose pretty early in animal evolution. So the same genes that control segmentation in one phylum seem to control segmentation in other phyla. So we know that those genes are homologous. In different lineages, those toolkit genes have been lost in some lineages, co-opted in other lineages, meaning they were originally used for one thing and now they're used for something else, or they were elaborated upon. So maybe you ended up with extra copies of those genes. So we ended up with convergent evolution of morphological segmentation. It says in. Anyway, so yeah, so the genes are the same, but then different things happen in different lineages. So you get both homologous and analogous things going on here that control segmentation. So just to reiterate how Hox genes work, so Hox genes are expressed along the length of the body, and if you make so Hox genes are like master controller genes that express affect the expression of the timing and rate of development of different parts of the body. So if you stimulate development over here with a Hox gene, you could end up with duplicated segments over here, longer segments, right? You could end up with a fusion of segments, and so basically a small change to a Hox gene can cause a large change to the body. And so then this is kind of like a really quick way to, if you have a small mutation, to have something that's very different in the body that is either advantageous in terms of natural selection or not, right? So then natural selection can favor variations that are helpful in adaptive environments, which causes diversification of different body types. So this is just kind of showing Hox genes that are expressed in the head of a millipede, also in a brine shrimp, grasshopper, and ant. The ones that are expressed in kind of like the little neck area. The millipede, nothing in the brine shrimp. Gives you the thorax in the grasshopper, none in the ant. The section that goes with the legs in the millipede is expressed throughout the body here on this brine shrimp, the legs here, and then the abdomen, very small in the millipede, much larger and longer in the brine shrimp, kind of crunched together and repeated in the grasshopper, and fused in the ant. Okay, so what kind of themes do you tend to see in the diversification of animals? So we know that most of the different kinds of body plans at the level of the phylum appeared during the Cambrian explosion. And this is one of the great mysteries in animal diversity as to why this happened. So why aren't we getting new, massively different body plans occurring all the time? Why aren't we finding really weird animals and you're like, is that its head? I mean, most animals kind of have predictable body plans that we've seen before in other animals. So one thought is that gene regulatory networks might limit the origin of new body plans. So the genes are interacting with other genes in this kind of a system, and once the system is established, it's quite resistant to change, right? But you can add to the network, so you can, if you don't touch the central part of the network, let me put it this way, if you have a mutation that messes up the network, then the animal probably dies. If you have a mutation that adds to the network but leaves the central functioning intact, then that will succeed. So as a result, you can modify the network, but it doesn't allow you to make a completely new network, so you don't end up with completely new body plans. All right, I think I mentioned this already in a lecture a little while ago, but remember we talked about this in terms of those non-mutually exclusive hypotheses for why animals were able to diversify in the Cambrian explosion. But just to remind you, one of the hypotheses was that because the oxygen levels were higher on Earth at that time, that might have allowed animals to become larger and more mobile because they had more efficient aerobic respiration. It could be that the evolution of predators at that point caused selection pressure on prey, and then the prey trying to avoid the predators exerted selection pressure on the predators, etc. The third non-mutually exclusive hypothesis is that once animals occupy new niches, that created niches for other animals in terms of other animals will try and eat that new animal or parasitize it or avoid being eaten by it, and then that created new niches. And then the fourth one had to do with those Hox genes. So as the animal genetic toolkit evolved, that allowed more potential for morphological diversity. All right, so just showing you some diversity amongst animals. So most animals can see, hear, taste, smell, and touch, or maybe not, but most animals can also sense temperature. Some have really cool specialized senses, so you have animals that can detect magnetic fields and use them in navigation, so like sea turtles can do this. Let's see, what else? So you have aquatic predators that detect electrical activity in prey muscles. So like sharks can detect electric fields. People have done experiments where they like bury live fish way down in the ground. Sharks can still find them. Birds can sense changes in barometric pressure. Actually, flies and stuff like that. A lot of insects can do that too. Other kinds of things. So some animals are stimulated by gravity. What else? Temperature. Pit vipers can detect temperature changes in their environment to figure out where prey items are. All right, there's also diversity in feeding. So a detritivore is an animal that eats dead organic matter. An herbivore eats plants or algae. A carnivore eats other plants. And an omnivore eats plants or animals. We say that a predator kills and consumes all or most of their prey. So it like basically just ends the prey item. We say an herbivore eats plant tissue without killing the entire organism. You know, usually like most things that eat plants will kind of eat the green part of the plant, but the root's still there and the plant can come back. A parasite will harvest nutrients from part of its host, but doesn't usually kill its host. And if you have a parasite that lives inside of the host, it's an endoparasite, and if it lives on the outside, it's an ectoparasite. All right, you got different ways of feeding. So you could be a suspension feeder, which is also known as a filter feeder. And so that could be like this little barnacle here, which if you've never looked up close, a barnacle is basically a little crab sitting on its back in this little volcano-shaped shell. And it just sweeps its legs, so it has these long jointed legs, and it sweeps them through the water, filtering out particles. But it's not just the barnacle that does this. Sponges, clams, baleen, whales do the same thing. Deposit feeders go along the bottom and kind of vacuum up detritus off the bottom. Earthworms also do this. Fluid feeders suck or mop up liquids like nectar or plant sap, blood, or fruit juice. So butterfly does this, as does vampire bat. And then mass feeders, that's like us. We eat chunks of food, like a lion or a snail. All right, which is which? So which one of these is a suspension feeder?
Oh, that barnacle over there, waving at me. Hi, barnacle. Hi, barnacle.
Yep. When's a fluid feeder?
The mosquito, yuck. It's wiggling its butt. You didn't see it get filled up with blood. Ew!
Yep, that's a fluid feeder. Who's a deposit feeder?
Well, who's that guy? I've never seen nothing like that. He looks like a pig.
He does look like a pig. I believe this is some kind of a sea cucumber, scrunching around on the ocean floor, mopping up detritus. And what about that last guy over there?
Well, he's a mass feeder, but I don't think he's supposed to eat noodles like that. I don't think noodles are good for crabs.
Yeah, I don't know. I'm not a crab, but I would bet probably not the healthiest thing to feed your pet crab. All right, in terms of movement, there are three basic types of skeletal systems. So a hydrostatic skeleton is a way of supporting a body without any kind of hard structure. So instead, you have a flexible body wall that surrounds an incompressible fluid or a soft tissue. So it's like having a water balloon. And so if you squeeze the water balloon around, you can make it long and skinny. And if you kind of flatten it between your hands, you can make it short and fat. And so basically, there are animals who can do that using muscle that surround, a coelom or some other unit of their body to be able to flex muscles against that trapped fluid. So that's a hydrostatic skeleton. So hydra means what?
It means water.
Yeah. An endoskeleton is when you have a skeleton on the inside of your body, like a vertebrate bone or a sponge spicule. Exoskeleton is outside of your body, right? Hey, let's ask a question here. So what's a turtle?
I mean, you've got a skeleton on the outside, but you've got a skeleton on the inside.
Yeah. Do you think a skeleton on the outside is actually structural?
No, because, like, you get soft-shell turtles and they fine like that.
Okay, so probably it's like extra protection, right? But their actual structure that keeps their body together is their bones on the inside of their body, right?
Oh, yeah.
If you've ever thought about it, the limbs, our arms and legs, are basically out pouches of our body wall.
Okay, that's gross.
There's lots of diversity in limbs, so you could have lobe-like hydrostatic limbs, so think of like a little caterpillar, or the lobe-like limbs of an onychophoran velvet worm. And so they're just little squishy limbs that it can move by flexing against the water inside of them. The little feet, tube feet of echinoderms, are also kind of hydrostatically controlled. The tentacles of mollusks are hydrostatic as well. However, the jointed limbs of arthropods, so the jointed limbs of an arthropod, is that an exoskeleton or endoskeleton?
That's an exoskeleton.
So where are the muscles?
I guess they're on the inside.
Yeah, the muscles are on the inside. So what kind of skeleton do you have, dog?
I got an endoskeleton. I can just feel it. Yeah, like here. Feel it. Okay, keep feeling it.
Okay, dog, I'm not going to pet you right now. We've got to keep going.
Oh, okay.
All right, so that's an endoskeleton. And so if you have an endoskeleton, the muscles are on the outside of that, right? All right. Traditionally, people thought that the jointed and unjointed animal limbs, so jointed meaning like an insect, and unjointed meaning something like a velvet worm, were not homologous, right? Because, one of them's got an exoskeleton, the other one's squishy, right? They don't look the same at all. And so people predicted that completely different genes would be responsible for making the two different kinds of repeated limbs, right? But more recently, we have found that a gene called distalis, I think it's DLL or DII, I can never quite figure out which that one is part of vertebrate limb formation. And so you can see that it's expressed repeatedly down the body of both an arthropod and an onychophoran, like this. And so that tells us there's genetic homology in how genes are expressed to produce animal appendages in both of these lineages. So, surprise, the limbs are very unlike each other, even though they repeat, right? They're structurally unalike. However, they're genetically homologous. All right, most animals reproduce sexually, but some can reproduce asexually. So, for example, like a rotifer can reproduce by mitosis. Some animals, even like big things like lizards or snakes, have been known to reproduce by parthenogenesis, meaning that they take one of their eggs and one of their, nope, I guess I'm trying to think how this works in lizards. I think it's just an unfertilized egg, isn't it? Or they fuse two eggs. I have to look this up. All right, somebody can look this up for me, or I will look it up and we'll find out. Anyway, asexual reproduction is not super common in animals, but the reason that there's selection for it is because it can be much more efficient, right? So if you reproduce asexually, your offspring are 100% related to you versus reproducing sexually, where your offspring are only half related to you. But the problem with making offspring that are genetically identical to you is that if you have a change in the environment, you have lower genetic diversity to be able to respond to environmental differences. And so you have animals that can reproduce either sexually or asexually depending on the environment. So, for example, something like an aphid will alternate between reproducing sexually or asexually depending on what time of year it is and how much food there is. All right. In terms of fertilization, there can be internal fertilization, meaning the sperm is kind of put into the female somehow and then fertilizes the egg inside of the female. There can be external fertilization, so the sperm and the eggs are laid outside of the body and then fertilization occurs in the environment. Some species are viviparous, meaning that they give birth to live young. So the species, the babies, the embryos develop in the female's body, and then she gives birth to live young. Some species are oviparous, meaning they lay eggs outside, and then the eggs will develop independently of the mother. So in order for those eggs to survive, the embryos have to have some kind of a yolk or something to live off of. And then here's a category you may not have heard of. Some species are ovoviviparous, so it means that the species will keep the eggs in the female's body early in development, but the mom isn't like kind of feeding them through a placenta or anything like that. Instead, the embryos are eating the yolk that's inside the egg. So basically what the mom is doing is protecting the offspring from predators, but the eggs are being nourished by the yolk that they already have with them. And then the mom can give birth to well-developed young later. All right, here's some weird shark facts. During mating, the male shark will insert the clasper into the female's oviduct and release sperm. This sperm then fertilizes several of her eggs. At this point, different things can happen in different species. So in some species, the sharks will lay their eggs. Some species, the sharks will nourish these young pups off of a placenta, or in some species, they will hatch the eggs within the oviduct before giving birth up to a year later. So, internal or external fertilization?
Internal.
Yeah, internal, right? All right, so so are these things ovivipary, or vivipary? So if the female lays her egg, what is that?
That's ovipary.
Yeah, that's ovipary. If the female nourishes the pups off of a placenta, what's that?
That's vivipary.
Yeah, vivipary. Or if she hatches the eggs within the oviduct before giving birth a year later, what is that?
I guess that's ovovivipary. That's kind of weird.
That's kind of weird. Hey, look, it's a picture of a two-headed shark.
Okay, that's real gross. Is that real?
Yeah, it's actually real. I mean, that's not how it's supposed to be, but apparently a shark can live with two heads. All right. In some species, you have indirect development, and that means that instead of the zygotes, the fertilized egg, growing up to be just like little mini adults, they grow into a different form called the larval form. And the larvae will look different from the adults, and they might live in a different habitat than the adults, and they might eat different food than the adults. And then at some point, they will metamorphose into juveniles, and at that stage, they look like little baby adults. So, for example, in a sea urchin, so the egg plus the sperm equals a zygote. Okay, the zygote will develop into a little tiny larvae, which will float around in the water until it lands somewhere, and then it'll metamorphose into a juvenile, which looks like a little tiny sea urchin, which will grow and grow into an adult. All right. So there can be advantages to indirect development. So if you are a species like a sea urchin where you don't walk around that much, it might be hard for you to put your offspring in a different environment, right? So by creating a larval form that is much more mobile than you are, it can travel much farther. The second thing is, if the larvae eat something different than the adults, then you're not competing with your own kids for resources. All right, so let's talk about what the fossil record and the phylogeny, the molecular phylogeny of animals agree upon. So they agree that Porifera, Ctenophora, and Cnidaria are the oldest animal lineages. Okay? They disagree about whether this one goes here or this one goes here, but we definitely agree that it's one of these, these three are the oldest. All three groups have a global marine distribution. So the Porifera here are diverse. They're sessile, meaning they don't move, mostly marine, meaning salt water, and suspension feeding. Some are predators, though. I actually found a little movie of some of the predators because I was curious about that, so you can see that in lab. So the adults are sessile, but the larvae will swim with cilia, and they reproduce asexually in a bunch of different ways. So basically, they'll all put out sperm and eggs, at the same time. We can tell them apart because they differ in size and shape, even in color, and they also have different types of spicules, which are those little those little skeletal elements. And they can host a wide range of photosynthetic organisms. So sometimes the photosynthetic organisms are what give the sponge its distinctive color. Sponges don't make this green stuff or anything like that. Just showing you, he put a little green dye on the outside to show you how it's sucked in the little pores out of the sponge and put out to the top just by the beating of the flagella. All right, ctenophora or comb jellies. There's not so many of them, fewer than 200 species. Most of them are planktonic and predatory, meaning they kind of, move in the water column, and they eat stuff. So you can't get stung by a comb jelly. They don't have stinging cells. They have kind of sticky cells, so they have a synapomorphy called a coloblast that's used to trap prey, but they don't hurt people. They're very efficient predators, though, but they don't hurt us. There's some very beautiful ctenophores. All right, so the Cnidaria include jellyfish, corals, anemones, and hydroids. So you can find cnidarians in all the world's oceans. Some of them are found like kind of in the surface. Some of them are all the way down in the ocean floor. Their main synapomorphy is the cnidocyte. So the cnidocyte is a barbed spear-like structure. It sits all coiled up until something touches it, and then it will shoot the little barb into your skin, right? And so the barb holds onto the prey, and then the toxins subdue it until it can be ingested. So the goal of the little, poison in the barb is to be able to catch little fish and things like that. But it hurts if it touches you, right? For a lot, depending on the toxin. So if you get stung by a jellyfish, it hurts you. If you touch a sea anemone, a lot of them, or most of them, don't actually hurt you if you touch them, but you can they feel sticky. And that's because they've actually barbed you in your fingers. And the barbs are so small, they don't hurt or anything, but it just seems like they're sticking to your hand because the barbs are in the top layer of your skin. All right. So jellyfish have a life cycle with both a sessile, so their stationary form is called a polyp. Looks like this is a polyp. And then the jellyfish-looking form is called a medusa. So they kind of have this life cycle where, you have jellyfish reproduce with each other, and, jellyfish produce a sperm and an egg, and then those come together to form a zygote. The zygote develops into a larval form that scrunches around on the ground and then forms a polyp, and so the polyp will get bigger and bigger, and eventually the polyp will get, and it's getting bigger by catching stuff with these little tentacles and eating it, and when it gets big enough, it'll produce this reproductive polyp here, and if you look at those little guys in there, those are each new little medusae that, when they get big enough, can get released. These ones are kind of coming off of a stalk. Some of them are really cute. It looks like a stack of cups. Anyway, so that's where you get these kind of medusa form of cnidarians. For something like an anemone and a coral, they only exist as polyps, and they don't have this medusa part of their life cycle. So a lot of the corals that we count on for building coral reefs are ecosystem engineers. So each tiny little polyp will secrete a calcium carbonate endoskeleton, and then when the polyp dies, the endoskeleton stays, so the structure is made up of bajillion little tiny polyps. Also, corals have mutualistic relationships with photosynthetic protists and green algae. So the coral itself will provide a place for the photosynthetic partner to live in. And so one thing that has been happening is that with global warming, this has caused the ocean to become acidified. And I think we go into this in 1113 a little bit, the kind of chemical equation that leads to this. And because a lot of the symbionts that undergo photosynthesis are very chemically sensitive, they may actually die, and so then the coral is bleached. So it's like the coral may still be there, but it lacks its symbiont, so it's unable to be productive. All right, so I hope you've learned what an animal is, the major innovations that occurred during the origin of animal phyla, different kinds of themes. We looked at the non-bilaterian animals, protostomes, and, we looked at the non-bilaterian animals and then mentioned what you're going to see in the protostome chapter next and the Deuterostome chapter after that.